Okay, so fish are animals and they respire, so they have a need for oxygen. So how they get oxygen is very different to us because they live in the water, so they cannot breathe. So where can they get their oxygen from? Well, the water there's a lot of oxygen dissolved in the water, so they need to manipulate that dissolved oxygen. They need to somehow come up with a way where the oxygen in the water can diffuse into their bloodstream. If it gets to their bloodstream, the blood can carry the oxygen to their cells, where it can diffuse into all the cells, and it can, respiration can then continue. So the first part, we'll talk about how fish get oxygen. We've got to just think about this idea of ventilation first. So we have our own version of ventilation, and ventilation is the ability to provide a fresh supply of something being oxygen rich. So we have oxygen rich air. How are they going to make sure they've got oxygen rich water? So we breathe in and out, and what fish do, they have to bring fresh supply of water to the gills. So the gills are found on the side here, and this is where they are pumping their blood to. So any oxygen that gets to this part of their body, it's going to diffuse into the gills, into the bloodstream, and then that oxygen can be used somewhere else. So to get a fresh supply of oxygen, oxygenated water, they open their mouth and they fill it up with water. They then close their mouth, and they've got a region here, this bottom part, it's called the buccal cavity. When they close their mouth, this bottom part is going to push up as well, a bit like a diaphragm in a way, being pushed up. And when that pushes up, the volume inside the mouth is going to get smaller. And if you remember from how we manipulate pressure in our lungs, if you decrease volume, you are going to increase the pressure. So where is that pressure going to go? Well, because they've got their mouth closed, the water can't go out the front. So the only place it can go, there's these tiny slits at the side of their heads, and it's something called the operculum. So the water is forced out this part here. And what's perfect about that is that's exactly where the gills are. So all this fresh oxygenated water is forced over the gills. Okay, so the oxygen needs to diffuse into the bloodstream. So we need the rate of diffusion to be as high as possible. So one way we can do that is by having a large surface area. So the gills, you've got a main gill arch here, and off this gill arch you get pairs of filaments all branching off it. And then if you go into more detail of these filaments, so if we zoom in, we can call these the primary lamellae as well, these filaments. And another way then to increase the surface area, each filament has these secondary lamellae coming off. And there's a better view of it here. So this is, you can imagine, this is one filament, like that. And that filament has these plates called lamellae coming off it. And they're serving to increase surface area. And these lamellae have their own blood supply. This is where the blood capillaries are going. So you can see here, there's an image of the blood capillaries. And the blue sides of the blood capillaries, so there's blood pumping to these lamellae, and it's bringing deoxygenated blood. This blood has already been used by the cells in the fish for respiration. And you can see as they move from one side of this plate to the other, they're becoming oxygenated, and they're moving back into the body where they can be used. Right, when water is forced through the side of the fish, its direction kind of twists a bit like this. Kind of takes that angle. So what's happened there is, if you imagine, you can see these blue lines here, you can see the water is being pulled at this angle. All right, and that's really useful because it's directing the water to go parallel with these lamellae. It's running through each of these little plates. Okay, so that's the direction the water goes through. It's not just running directly over the top here. It's moving through all of these filaments in line with all of these secondary lamellae. Okay, so that's good for increasing surface area. So the more surface area you have, the more opportunities there are for diffusion to take place. Now the next part that the fish have got to maximize diffusion as well is they have got a way to ensure there's always a high concentration gradients. So if we talk about, let's go back here actually. If we talk about diffusion will always be at its highest rate if there's a high concentration gradient. So if you've got an area where there's loads and loads of, let's make this a bit bigger. If you've got an area where there's lots of particles, let's say oxygen, compared to an area where there's not many, this is going to diffuse at high rates. As it starts to diffuse though, it starts to move over, and you get more and more particles over here. Eventually it will equilibrate and diffusion will stop. So you don't want that to happen. So what a system has a fish got to help to try to prevent the concentration gradient from getting too small? We want it to be nice. We want it to be nice and steep all the time, as steep as possible. We want high concentration, low concentration. We do not want the concentration gradient to look like this. Yeah, that's going to cause diffusion to slow down. So I'm going to zoom in a bit more. So these plates now we're going to focus a bit more on. So here's another image. You can see the water goes into fish's mouth. When it opens it, it fills it up with as much water as possible. The buccal cavity, this region here, the mouth is going to close, and the buccal cavity is going to push up like a diaphragm, causing the volume to get much smaller. Therefore, the pressure is going to increase, forcing water over the gills. And the water is going to be, let's make it a bit smaller again. The water is going to be directed, as it moves over, it's going to start moving this direction. So it's going to follow the line of all of these lamellae here. Okay, so these lamellae, or individually called a lamella, this is where gas exchange takes place. This is where the blood is going to. So the blue line is where the blood's entering, deoxygenated, and the red is where it's leaving. So you can see the water is moving this way, but the blood is moving this way. We say that it's countercurrents. So we're going to talk about this now. Countercurrent gas exchange and how it maintains a concentration gradient. So let's have a look. What would happen if the blood and the water moved in the same direction? You would still get gas exchange, but it wouldn't be as effective. So let's look at this in this example here. Let's imagine the water and the blood, as blood cells are moving together. So, although it wouldn't be 100%, just use these numbers to make it a bit easier. Let's imagine the water contains 100% dissolved oxygen. So, that's really high in oxygen. And this blood cell has just come from the body, so it's been used for respiration, the oxygen, and it's got no oxygen at all, it's at 0%. So clearly, it's going to get diffusion happening, because there's a high and there's a low. That's really good, it's a really steep concentration gradient. So as it starts to lose oxygen to this red blood cell, well, because they're moving together now, it's down to 80, it's given it 20, so this diffusion can still occur. Gets down to 70. Already, because this is starting to go down to 70 and this is going to 30, the concentration gradient is less. There's only a 40% difference between them. So the diffusion is starting to slow down. It's gone from 100 down to 80%, down to 70, and now it's only at 65%. Diffusion is still happening, but it's starting to slow down. So by the time this red blood cell has come into the lamellae, remember it's coming in through here and leaves, it's only picked up 40% of the oxygen that it might have potentially been able to pick up. So it's now going to take that back to the body. So yeah, you'll get some oxygen, but not as much as you could if you went countercurrent. So this is what the fish has got set up. So rather than the blood going this direction, it's going countercurrent to the movement of the water. So this oxygen here, where it enters, is 100% rich in oxygen. And by the time it's given up its oxygen, it's down to 20%. When it's down to 20%, a new blood cell has just arrived from the body. So this is coming from the body. Even though the oxygen in the water is only at 20%, because it's still much higher than 0%, diffusion can occur. And as this blood cell moves through the lamella, up to 20% now, it's now hitting a region of water which has slightly more oxygen, so it can still get more. There's still a concentration gradient of 50% because the water has 60, so it can still get more. And as it goes up to 70, well, because we're now at the part of the lamella where fresh water has just arrived, this water's at 80%, so it can still get oxygen. And even as it gets even closer to the end, it's still picking up oxygen because now this region of water just here that's entered is at 100%. So this blood cell is leaving with 90% oxygen. So significantly more oxygen being carried by this red blood cell. So the effort that the fish has made to pump this red blood cell to the gills and then to return back, well, it's achieved a much greater amount of oxygen. Okay, so a quick summary. So different parts of this. Reminder, fish need oxygen just like we do. They don't get their oxygen by breathing. They get it by forcing water over their gills, and the water contains dissolved oxygen. That oxygen in the water needs to diffuse across the walls of the lamellae to get into the blood. Diffusion is only going to be really effective if we can maintain a steep concentration gradient, and that steep concentration gradient is being achieved by blood moving countercurrent to the water. Gas exchange surface area. This has been made as maximum as possible by the structure of the gills. We have individual gill filaments, and then on each gill filament we have these lamellae which are massively increasing it, and that's where the blood is being pumped to.